Yeah, sorry, Priya, I just had a call.
Okay.
Yeah, I think we are good to go. Is it not, Tuhina? Shall we start?
Yes, sir. So, welcome back to the 8th International Conference on Biosignals, Images and Instrumentation for the year 2022. To begin with the final day's keynote session this afternoon, I would like to extend a warm welcome to our first keynote speaker, Dr. Priya Lakshmi Narayanan from Institute of Cancer Research, London, United Kingdom. Dr. Priya Lakshmi Narayanan received her PhD degree in biomedical engineering for her work in developing an atlas of cerebellum for young children. She worked as a research scholar in MRC or UCT, Medical Image Research Unit, University of Cape Town, South Africa from 2010 to 2015. She has also worked as a visiting academic in Athinola Amartino Center for Biomedical Imaging in Boston from July 2012 to December 2012. She has gained a considerable amount of experience working as an image analyst and deep neural network based framework development and its relevance for validation in breast cancer. Her expertise in image segmentation and cellular phenotype quantification, along with complex data analysis and interpretation, has resulted in significant research outputs in Nature Breast Cancer and Nature Scientific Reports. She won a Marie Curie Research Fellowship and moved to the UK during her final year PhD research program. During her tenure at Path Excel, UK, she has been an active part of the patent for which she is recognized as an innovator while at Path Excel in Fast Path project. She is actively working in understanding recurrence pathways and resistance mechanisms in breast cancer with Institute of Cancer Research for the past 4.5 years. Currently at UCL, she works as a research fellow. She is an active member of Department of Computer Science in building intelligent systems that help to predict acute myeloid leukemia. She initially started her career from a startup based in India, Optra Systems, Pune, after completing her master's in 2007 from Madras Institute of Technology, Anna University, Chennai, India. During her tenure, she contributed to the detection of lumen in prostate cancer and microarray analysis for single cell detection. We welcome you, ma'am. Please take over the session now.
Yeah, thank you. Good afternoon, everyone. I'm happy to be part of the International Conference on Biosignals and Image Analysis today. Um, so, so are you all able to see my screen? So can I continue? Yeah, so today my talk is on breast pathology and the hallmarks of computational evolution. So from the COVID-19 experience in the recent 2 years, we all know the cell surface has receptors. They have multiple receptors, and in cancer domain, the cancer cell and the immune cell handshake, and they are through these receptors and the presence of molecules uh, actually the presence of molecule actually responds to the treatment. So this treatment improves the patient survival. So this is the mechanism that is being used in the drug development scenario. So with this as a background, let me take you to my presentation outline. So it is going to have some background about the domain, and then I'm going to introduce to the biological hallmarks in cancer development life cycle and the computational use cases that I have contributed in the development of unmasked and PTM network development, and then the systematic web interface demo that I'll be giving as a final part of my presentation. So why we need computational pathology? So because all the biopsy specimen that reaches the pathologist is examined under microscope, and this remains as a gold standard for diagnosis in cancer. But this is subjective, so that we need a quantitative measure that can be used in the validation. So in recent days, there is improved scanning and storing and algorithm development that has gained significant momentum, which has a direct implication in translating these computational tools to assist the clinical translation. So the computational pathology and the tools helps to mine richer information from the tissue images, which is not possible through the naked eye. Because while a pathologist seeing a tissue under a microscope is restricted to the number of field of view, so that the evaluation can be subjective. So he analyzes only 5 or to 10 field of view because it increases the fatigue and it reduces the interoperability and inter-rater variability between the pathologists increases. So that's why there is a huge need of a computational tool that can reliably scale up the processing of the whole slide images. So let me throw some biological background initially. So for cancer, it is a complex ecosystem which has heterogeneous phenotypes of cells, which accumulates over a period of time. So in here, you'll find a red cell that is a founder cell. We call it as a founder cell. So these red cells get accumulated and generates another phenotype. So, and after a genomic event, either a mutation event or a copy number alteration will cause the phenotype to change, and these get accumulated. So likewise, there are cell phenotypes that gets accumulated over a period of time and turns a particular phenotype to be a dormant cell. The dormant cell is nothing but a cell that that never dies and sustains the cancer evolving in the patient's body. So the main biological hallmarks are these 6 hallmarks in cancer, where there is proliferative signaling. Proliferative meaning uncontrolled growth. So uncontrolled growth happens when there is cancer. So it evolves and then evades the immune mechanism and the growth suppresses, which in turn activates the invasion and metastasis in a patient. So what happens is when it actually accumulates these cells in one place, it introduces replicative stress inside the tumor. And this creates an ecosystem for which there should be a blood supply, and then there should be the transport of waste materials, which is helped by the angiogenesis process. And then there is this dormant cell mechanism, which resists to make the cancer cell die. So these are the 6 biological hallmarks that are in cancer. And I'm going to tell you how we are going to use one of these to solve quantitatively and computationally. And there are molecular subtypes of breast cancer, wherein on the left-hand side, if a patient is diagnosed with luminal A or a luminal B subtype, they are in a better prognosis category because these patients survive longer if they fall in luminal A or a luminal B subtype. The luminal A and luminal B subtype are those where there is this estrogen receptor that is positive, which is favorable for the patient survival. Patient survival means how long a patient can survive after being diagnosed with that particular type of breast cancer. And the opposite spectrum of triple negative is where this estrogen or progesterone receptor is not present. So they're all negative. So that subtype is called as triple negative. That is the worst subtype because it's quite heterogeneous. And right now, there's not many drugs that can help these patients. So they fall under the spectrum of worst prognosis. As in, like we were thinking COVID was the worst prognosis subtype, but because of introduction of the immunization schedule, so it has become into good prognosis right now. So you can relate these terminologies associated with COVID. So the challenges in developing a multimodal image analytic framework is you need to know about what are the different tissue levels you can use in the whole slide image analysis. The whole slide image is a gigapixel image wherein the pixel size is 1 lakh pixels by 80,000 pixels. Just imagine how much computational power and computational processing would it require to address the cell level characteristics and classify the individual cell phenotypes. So there are different staining as well. The one on the left is the hematoxylin and eosin stain, which is diagnostically used everywhere in the lab throughout the world. And there are immunohistochemical stain, which are in the middle panel, so which are stained for specific marker, which I said in breast cancer, there are 3 different markers, that is ER, PR, and HER2. So they check once they are once a patient is diagnosed with breast cancer, they would stain for ER and see if a patient is ER positive. So which means that if this particular protein is expressed in the cancer cell, then it becomes ER positive. So if a cell is positive for that protein, then in immunohistochemistry, it gives a brown signal on that particular cell. So that's what happens in the immunohistochemistry. And this and the same way for progesterone, it will be the PR marker. So in the same way for the proliferation, there is a marker called Ki67, which quantifies the uncontrollable growth. So you stain for Ki67 and the nuclear expression will be obtained in the cell. And then those are quantified computationally. And there's the other marker, which is on the right panel. I don't know if you can see it. So these are the multiplex panels where, because you wanted to study the whole tumor microenvironment, meaning you wanted to study cancer as well as the surrounding cells. You mean there are a number of immune cells that are present in the complex ecosystems. So which means you need not only one marker, you need 2 or 3 markers. So to study that, there is a technology called multiplex immunohistochemistry stain. So there are 3 stains, more than 3 stains. In immunohistochemistry, there is only 2 stains, but in multiplex immunochemistry, there are more than 3 stains. So let's stick to the hematoxylin and eosin images and stick to the morphological growth patterns that occurs in breast cancer. So normal breast duct have a regular columnar epithelium, which is surrounded by the basement membrane. So this is the normal duct. Whenever there is atypical growth and there are more columnar epithelium that gets populated inside the duct, they are called as hyperplasia. So this atypical growth, once it grows profoundly, then it's called as atypical ductal hyperplasia. And then once, because whenever the cells accumulate, when there is uncontrolled growth, there is a formation of a lumen and there is a secretion of necrosis. Those that are an yellow or like a necrosis pattern that occurs within the DCIS. Wherein in this, you can see my mouse pointer, I guess. So wherein I'm pointing to the rightmost figure. So wherein there is a necrosis in this center. So what happens is this creates a stress, and then there is a signal to break out this basement membrane. Once this basement membrane is broken down, then it becomes invasive. So this is the ductal carcinoma in situ, where it is indolent, which means it resides inside the circular pattern. When it invades the basement membrane, that's when it becomes invasive breast cancer. So then you call that type of cancer as a malignant. So, and it forms an invasive pattern. So invasive will form a irregular pattern, and that causes more lethality to the patient. So for the whole slide images, because I said 1 lakh pixels by 80,000 pixels, just imagine how many tiles. So if you are separating in each of these region into 2000 by 2000, just imagine how many tiles you will get. So let's say we are getting 8,000 tiles. from the 1 lakh pixels when you divide it by 2000 by 2000 pixels. So in the lowermost layer, you will get the number of tiles to be very high. As you grow up on the pyramid, you will get less number of tiles. So usually in this pyramid structure, it is stored in such a way that it is 1:1 and 1:2 and 1:4, 1:8 and 1:16. So that way you can access the low resolution image from the 1x resolution. And if you want to go to the 20x, you will get more number of tiles for the processing. So this is one of the challenges that needs to be kept in mind because you need to capture all the heterogeneity associated with one particular patient. And on the right, you can see that this is a theoretical explanation of how a normal duct can grow into invasive. These are very good figures and explanation, but in real world, this is a scenario. It never is, a duct is never being a perfect round circular region. It appears in different patterns. So these are the heterogeneities that needs to be captured when you wanted to train a deep neural network. And now I'm going to introduce the unmask. So wherein the aim was to develop a computational tool to enable morphological analysis and then study the differential immune ecology in order to identify a particular score that can actually stratify the patient who are lethal in a given subgroup. So for unmask, what did we do? So we saw the challenge in 3 different levels. So one at the tissue level, then at the region level, and then we computed the spatial ecology. How did we do that? So what we did is, if you remember the pyramid structure, we extracted the topmost resolution, which is a low resolution, which is a 1x of that image from where it is a bird's eye view of that whole slide. And then we identified the foreground and the background so that it reduces the number of tiles that will be available for the computational process. Let's say there are 8,000 tiles. Because you are finding these are the foreground, you would restrict it to process only 2,500 tiles for the high resolution tiles. So that it reduces the amount of time that is required to process all the whole slide image associated with one patient. So then what you do is you segment the DCIS at 20x, and then you identify them at 20x. And then once you identify all the DCIS and process all the tiles, what you do is you segment the cells at the single cell resolution. So once you segment the cells, you identify and classify each of these cells and classify each of these cells, because in a whole slide on a H&E, there are 4 types of cells that can exist, which can be reliably quantified. So those are stromal cells, immune cells, and the tumor cells. And then what we do is once you get the cell phenotypes, you now have the centers of the DCIS. You put the centers of the DCIS and then calculate the Voronoi tessellation. So you estimate the Voronoi. And then what you will be able to do is you will be automatically able to define the tumor microenvironment that can be associated with the individual duct. So what we inferred is when we did the Morishita score, when we calculated the Morishita score is nothing but we can associate 2 different cell phenotypes using this Morishita score. You can look up my literature of how the Morishita score is defined and calculated. And what we observed here is whenever the immune is depleted, the Morishita score tends to be less. And whenever there is highly inflamed duct, wherein the immune cells are quite high and richly distributed, the Morishita score was high in those cases. So then we calculated the overall average for individual patient of how this Morishita score behaves in pure and as well as in the adjacent DCIS category. So highlighting the methods, what we used is encoder-decoder based mechanisms wherein we had the ground truth as well as the image given to the network. We use a distance map in order to regularize the network, and we obtain the segmentation of the DCIS. And then for the individual cell-based detection, we use the cell coordinates, which are X and Y positions of individual cells, which are in the annotation. And then we use the Huff-Stroke distance to regularize our network. So that way we were able to identify the individual cells. So now after classification, after the classification of cell phenotypes, we estimate the Voronoi because the DCIS is segmented. From the segmented DCIS, we are able to extract the centroids of individual DCIS. From the centroids of individual DCIS projected onto the low resolution image. Again, we come to the 1x image and we project the centroids onto the 1x image. Because they are saved in a pyramidal format, we can actually bring back each of these coordinates into the lower resolution. Once the centroids are plotted on the 1x image, we can estimate the Voronoi, which decides the tumor microenvironments. Once that is got, you associate and get all the coordinates in the higher resolution to the lower resolution, and then you compute the Morishita score. So once you compute that Morishita score, so what we did is we calculated the percentage of lymphocyte for each of these patients in two of these groups. One is pure DCIS group and the other is the adjacent DCIS group. What we found is the pure DCIS, although the percentage of lymphocytes seem to be high, it doesn't represent that your Morishita can be high. So what we found is in the lethal subgroup of adjacent DCIS, because of the co-localization that is happening within the cancer cell and the immune cell, it was able to become a lethal phenotype. This was an interesting finding. And because of that, we wanted to know what are the type of immune cells that are present in the vicinity of the DCIS, which meaning what is the type of immune cells that are present in the vicinity of the DCIS. So there are different type of immune cells. There are CD4, CD8, and FOXP3, which are popularly used to see whether they are active, which can contribute to the invasion or the evasion mechanism of the tumor microenvironment. So what we found is CD8 is a phenotype that was present more when compared to the CD4 and FOXP3 phenotype, and that was significantly more within the DCIS, because we have the Voronoi estimation. From the Voronoi estimation, we can segregate the invasive region and the in situ region. So from where what we figured out is that within the DCIS region, the CD8 was quite high, so which is causing the Morishita value to go high. So we know that now these are the phenotypes that are actually driving the lethal phenotype. So the next one is the multiplex immunohistochemistry framework. So wherein as I said, there are three different markers. So where the CD8 is in pink color, where the galactin-8 is brown, which is a tumor cell, and P-stat, which also stains the tumor cells. So it's the same cell stained by two different markers, and there's also this immune cell, which is a CD8 positive. So what we are studying here is these are ER negative samples, which means the estrogen receptor is not expressed in these patients, so that they fall into the ER negative category. And the spatial immune cells distribution also differs in this ER negative category. So what we wanted to do is we wanted to develop the single cell spatial mapping so that we can study the spatial heterogeneity of the immune cell, which leads to the immune suppression. So what we inferred here is wherever we found the galactin-8 was high, there was less CD8 infiltration. And the spatial interplay between the galactin-8 tumor and the CD8 T cells depend on the P-stat expression. So as you can see, the blue color, the P-stat, we had 3 intensity profile, which they call it as weak, moderate, and strong. So for the data preparation, again, it's all the cell coordinates that's been annotated into 5 different categories. One is the CD8 positive. Then the next is a galactin positive and P-stat negative. And galactin positive, P-stat positive, weak, and moderate and strong, depending on the intensity profile. So for the classical deep learning problem, we used the stratification of 70% training, 15 validation, and 15% testing. So what we did here is we used the same encoder decoder based network. So wherein 224 by 224 by 3 is the input patch given to the network, because here we had a challenge because it's a very low intensity. So that what happens is the number of cell detection that we obtained were higher when compared to the normal H&E image. In the multiplex immunohistochemistry, because of the very light intensity that needs to be captured, so we got more number of cell detections from the patch than the desired number of cell detection. We got more number of cell detection when we use the encoder decoder network. So what we did is we parallelly used a cell counter network, so wherein we regularized for each patch how much cell count is present. So depending on that, we regularize the network to get the optimum number of cell counts per patch, so that we could overcome the challenge of the low intensity problem and the false positive that can occur from individual patches. So for this, we use the dice overlap loss and the cell count loss that was used for this cell counter model. With this, we regularize this network and then post-process to obtain the final cell detections. Once these cell detections are obtained, so first classifier, we had CD8 positive and galactin 8 positive and p-stat negative, and galactin 8 positive and p-stat positive separately. And then once we design our first classifier, we went on and then we classified this one into the next classifier. We fed this input to the next classifier in order to separate the intensity profile, because that was a challenging thing to give to the one classifier as a to identify the 5 different classes. So we gave the third class to the second classifier in order to identify this weak intensity profile, where we could classify the p-stat moderate, p-stat weak, and p-stat strong. So these are the results that came from the cell detection. So wherein in the concordinate, we obtained a higher recall and a higher FN score rather than the traditional unit or the only without the cell counter. The precision was high, but the recall was not that high. So introducing the cell counter and the inception architecture actually helped to achieve better recall and better reference score in our cell classification. So these are the high resolution tile where you could see all the cells where the CD8 are actually classified into like white dots. And once you obtain all the cell phenotypes, what we do is we extract all the coordinates and then put it on the 1x image. So if you put it on the 1x image, you're able to now see the different type of lymphocytes. So with respect to, once you get the CD8 positive, you classify all of the CD8 positive into adjacent tumor lymphocyte, intratumor lymphocyte, and distal tumor lymphocyte. So you classify them again, and then you calculate the intertumor lymphocytic ratio. And what we found is there is a negative correlation between the strong galactin 8 expression and the intertumor lymphocytes. So which means that wherever you have higher galactin 8 expression, there is a low concentration of CD8 cells, which means that there is this spatial heterogeneity pattern, which is quite different in the ER negative breast cancer. And so we also wanted to see, does the spatial interplay between the galactin and CD8 depend on the p-stat expression? So because we have p-stat weak, strong, and moderate, so what we could see is the CD8 distance with the p-stat weak, p-stat moderate, and p-stat strong. So what we found is the p-stat expression, which is strong, was having less distance between the CD8. So which means that the strong p-stat expressing tumor cells were closer to the CD8 cells than the other p-stat weak or the p-stat moderate. which shows the spatial interplay of the CD8 is dependent on the p-stat expression. And then to the next publication, which is based on the proliferation tumor network. So this is based on the Ki67 marker used for proliferation. So what we did is we annotated the whole slides and extracted the regions of tumor and the non-tumor. So once you do these annotation, so the whole slide, because it's of 1 lakh pixels by 80,000 pixels, but by annotating a small region, you would get the regions of like around 1,000 regions for a tumor and 1,000 regions for a non-tumor. So likewise, if you have 10 images, you would get around 10,000 images that will be readily available for the training. So this was used to derive the tumor and the non-tumor segmentation, so that can help, because in a proliferation, it is very important to know that you only score within the tumor region. So you don't score within the non-tumor region. So once the tumor region is identified, it is easier for the pathologist to then score only the regions within the tumor region. So that is why this becomes an important problem to identify the tumor region. Otherwise, they would go and search in a non-tumor region. So how we quantify this? So this is a whole slide where I'm showing two different areas of interest. So these are the high resolution image, and these are the high level of annotation. So if you are drawing these annotation in a low resolution, you cannot, these boundaries will not be binding to the tumor region. They will be more distant from the borders of the tumor. Because it is drawn on a high resolution, that's why it is sticking to the boundary of the tumor. So because you could always use a low resolution and then try to extract the patches and then do the analysis, but that would lead to the suboptimal results. So it is very important to get the high level of annotation, but doing a high level of annotation will take a lot of time. So for one slide, it might take more than 3 hours, so that you could expect how you're like, if you want finer results on the tumor analysis output, then you have to spend this time on the annotation, getting this detailed annotation bounding to the tumor border, because that's more important, because any low resolution can result in a suboptimal result, because the tumor borders are important. Because here in this figure, there's closely lymphocytes which are next to the tumor border. So if you're going to train with the low detailed annotation, you're going to include the lymphocytes region into the tumor region, which is not acceptable by the pathologist. So we did the high level of annotation and then trained the regions and the patches involving within the regions, which actually resulted in a better DICE score. So because now we have the ground truth annotation, we are able to map them or the predicted annotation on top of it. And those that are in green are the true positives, because you can overlay the ground truth and the predicted map one on top of the other. Those pixels that are green are the true positive pixels. Those pixels that are pink are the false negative. And those that are in yellow on this image is the false positive pixels. And those that are in blue are the true negative image. So once you obtain this for the high resolution, these are the activation maps for the tumor region. So you could see the level of detailed annotation resulted in a very good activation map, which bounded to the tumor borders. So because these borders cannot, so if you see in the manual annotation, these borders were not annotated, but because you have done a very good job with the annotation, this tumor map was able to segregate those smaller regions or the stromal regions that are within the tumor. So which results in a better DICE score. So DICE score is nothing but the spatial overlap between the manual annotation and the ground truth annotation. The intersection over the union is the DICE score that is computed. And this is a part of the patent that I was involved while at PathXL. And I'm the inventor in that patent. So wherein there is stain normalization method that's been incorporated along with the artifact correction that can occur in the whole slide image analysis. So let's say like how the analysis can mediate to personalized therapy and how can it guide a pathologist. So what can be done when you're given with a H&E slide? So what can you compute? So these information are readily available on data sets like TCGA. So TCGA has opened up all the whole slide images along with the mutation, along with the therapy details that are given to breast cancer, given to lung cancer and everything. So these information can be extracted and can be done into a data analytic framework. You can systematically develop the cell segmentation, cell classification, tissue detection, and the regional analysis, and combine these with the data analytic framework that you can extract from the TCGA, depending on the genes or with respect to the specific therapy that is given to the particular patient, and associate individual features that you can extract from the deep learning. And then you can associate that with the patient outcome and see what inference you can get from the by combining the integrative analysis. So there are complex challenges when you go with the whole slide images. TCGA has images from the Aperio whole slide scanner, but there are other whole slide scanners such as NDPI, and NDPI and Philips, and there are many other scanners. So these, when the scanners are different, you need to make sure that the deep learning model gives the reliable results. So how we can test that is if it's possible, the same image can be scanned on Aperio as well as a Hamamatsu and a Philips image, and then see how a model performs. That actually validates your model, and you need to do that. And then you also need to do in all of your pipelines, such as cell detection, cell classification, and everything. So it's all been done on the unmasked. All these validations were done with different scanner settings, different pipeline, and all of them were validated with 2 different scanner settings. And then you compute those features and you use those features in order to segregate the patients into 2 different categories, like who responds to the treatment and who doesn't respond to the treatment. So these features can be agnostically derived from a deep learning based feature. But it is very important that how you integrate the data and how you interpret the data. So it's very important that the data analytic framework should go hand in hand with the image analytic framework and the feature deriving framework. And there are multiple modes in which this can be done. So this is the web viewer that I have been developing over the years in order to handle the whole slide images. So you could see that how much information is there from individual resolution. So these are regions of invaded breast cancer, which is invasive breast cancer. So which is not a regular duct. There's so many duct which has formed an irregular pattern. So let me show you so how I would be testing my classification results on the whole slide is by laying the original image and the classified output side by side. And I would zoom in on to every region and see how the classification results look like. And you also estimate that based on the XY coordinates that is present in the ground truth as well as in the predicted coordinates. But this is a visual quantification of how scalable the analysis can be. because it's a high resolution slide, it doesn't pop up with the memory that is in the background. So I also wanted to show what are the problems that can occur. So that's why I'm showing the region when it was not able to show the synchronized view of the image as well as the overlay. So the one on the left is the original image, one on the right is the classification output on the P67 marker. So this image can easily have 4,000 tiles. Yeah. So in order to get the annotation, so there are tools for annotations. So you select what type of annotation. So in this framework, you will be able to edit the annotation and mark the annotation. You can select them as a square or a rectangle and then make your annotation. And these coordinates are used to extract the patch information for your deep learning. So there is a whole pipeline that needs to be there before you think about a network and implement the network. So this shows the scalability and the achievability of reliable quantitative metric that we can achieve through the digital whole slide computational processing. And the Unmask framework, all my pipelines are available online on GitHub. And then you can also, it's also available as Docker images. So if you're using Docker, then you can use my model and then test it on a whole slide image and see how well individual pipeline works. So there is separate pipeline for the tissue segmentation, separate pipeline for DCI segmentation, and there is separate pipeline for H&E cell detection and H&E cell classification. All these codes are available on my GitHub, and you can refer to my GitHub and my publication to go and if you are interested to carry out some initial sanity checks of how this pipeline can be implemented step by step. And that's it for all today. And thank you. Hello? Do you have any questions from your side? Hello?
Yes, ma'am. One second, ma'am. I'll ask the participants to ask for the question.
Sure.
Well, just a minute.
Participants, if you have any questions, kindly post it in the chat box. Okay. If the participants do not have questions, we'll move forward. Thank you, ma'am, for your detailed presentation. We are sure that the audience interested in exploring and carrying out research in evaluating the evolutionary trajectory in breast cancer would have been much benefited by the detailed explanation that you have provided. Thank you so much for joining us today. We are happy to have you. Thank you, ma'am.
Okay, thank you.
Participants kindly be on the same link till Dr. Bello joins us. Welcome back, everyone. Now we have with us our last speaker for the keynote session. I would like to welcome Dr. David Bello from Fraunhofer, Portugal, AICOS, Portugal. He is a biomedical engineer with a vast and multidisciplinary experience. Dr. David Bello has received his PhD in biomedical engineering from the Nova University of Lisbon in 2021 in applying deep learning algorithms for several biosignal paradigms. His research activity was mainly in biosignal processing and machine learning to extract meaningful information and make automatic decision support system. And on the industry side, he earned experience in programming mobile applications, reporting services, and administration software. His specialization includes deep neural networks, TensorFlow, Theano, biosignal processing, machine learning. In addition to this, he has participated in the summer of 2019 in NASA Frontier Development Lab in developing AI tools for aiding the astronaut health program. We are glad to have you, sir. I welcome you to begin the session.
Good morning. I'm sorry for this brief moment. I was trying to unmute myself, but I wasn't able. Thank you for receiving me. I will now share my screen. So thank you for having me. I'm very excited to present to you my presentation. Here in Portugal, it's still in morning, so sorry if sometimes I'm a little bit slow as I just had my coffee. So I'm a senior scientist at Fraunhofer ICBSII currently. And I wanted to give an overview of my work that I did on using deep learning architectures in the biosignal context. So, what is the motivation? Healthcare as a driving force. As you may know, it's one of the most powerful factors in empowering well-being, especially on these days due to the pandemics. This is now, all of us are very aligned with this concept, right? Because it has been tough years, and we really know that without health, we don't have functioning systems in terms of as a society, but also as for us individuals, it's very important for us to retain our health and be healthy and be to feel good about ourselves and to live our lives. Currently, there are a lot. The technology in healthcare, it's expanding exponentially. And we can see also again, so the COVID-19 was very demanding and we actually got a vaccine in due time, and the research has grown exponentially. This is very well seen, right? So everything is, the technology is in terms of diagnostics, therapeutics, and innovative technology. It's it's increasing in a very, very quick way. Also, the medical health records has increased medical practitioners' accessibility to patient data, right? So once we are in the hospital, it's easier to gather information from other departments, such as the surgery department, the X-rays, and etc. we can access that information, not us, but the medicals, since there are privacy and anonymization issues. But as a practitioner in my hospital, normally I have access to data in an electronic way. So it's easier to access this data. That has been here, it's more focused on the European Union since I am in Portugal, but in the world it has been, there has been an investment increase on the healthcare data and the healthcare technologies. Also, the accessibility to wearables. So the record of human physiology activity in the past was confined in the healthcare facilities. We couldn't get any kind of data in 10 years, 15 years. We didn't have access to data other than outside the practitioners and the healthcare centers. But currently we can get a watch that can monitor our health signals. We also have our mobile that can get our activity, and with that infer about sleep patterns and etc. and activity. So there has been a digital revolution ever since, and there are a lot of wearable devices, more types of data that we can get. There are more sensing devices, and they are smaller and less invasive over time. So there is a lot of data acquisition. This has two edges. So the first one is that we have a lot of data that we can analyze, but at the same time, we have a lot of data that we have to analyze. So as this quantity of information increases, it's also very difficult to analyze it. So also part of the motivation is the recent rise of artificial intelligence. So artificial intelligence breakthrough, mainly boosted by deep neural networks, commonly known as deep learning technologies, has been a branch of computer science developed to mimic and help human intelligence. There is a lot of transformation potential, also economical potential, and especially for healthcare applications. So as we increase this amount of data, and also the industry and also the academic have gained more trust in adopting these deep learning technologies, with results that they are shown and they have been proven over and over again. We can state that this is a wonderful technology to use when using this kind of data, as it's increasing exponentially. So the motivation behind this is actually the mixture of these three domains. So this presentation provides several deep learning architecture for a wide range of biosignals and objectives. This was done during my PhD. Most of it was during my PhD dissertation. And some of these architectures were benchmarked in three different contexts: in synthesis, authentication, and detection. And I hope this study of these algorithms helps unveiling new knowledge about the intrinsic nature of biosignals. Okay, so if is it possible if these networks learn patterns that we as human cannot perceive? Will it be possible to understand a little bit more about the nature of the biosignals, even if it is for a little bit? So I will, this will be the overview. Well, I'll give you a little bit of context. Also, some methods for biosignal processing for deep learning architectures, the applications that I mentioned you about, some conclusion, future remarks, the literature, and I will give you also a brief presentation on the internship that I had in NASA, and then the references. So by starting, what are biosignals? So I can say that the biosignals are originated by the physiological process of living beings that can be measured and monitored, considered by appropriate sensors. So as we put sensors in our body, we can gather information about what is happening, what is the electrophysiological, or what is the motion that is happen within the place where the sensors are put in, right? So if we have an ECG, then we have a signal that is acquired, and all the phases of the signal represents the state of the organ that we are measuring. For example, the EEG, we have brain waves that look quite random, but as we study it more carefully, we can understand there are actually patterns, namely on the different frequency bands of the signal. The EMG, we can see, for example, the activation of the muscle where the electrodes are put in. But not only electrophysiological signals are gathered, but also mechanical ones, such as the respiratory systems, where the band, where the signal reflects our respiration, so the enlargement and also the compression of our thoracic cage. We can say that the artificial neuron is a representation of the real neuron, where we have here a representation of the neuron and also a trigger. So the idea is that as the input of the neuron is stimulated by a neurochemical or electrical discharge, there is the voltage of the soma will increase, and after passing a certain threshold, then the activation threshold is achieved, and then we have an action potential. So to mimic that conditions in terms of a simpler way, we have the artificial neurons where we have several inputs. Each one has its own weight and also a bias. And we can say that this bias, the B, is actually like an activational a threshold, because after passing through the activation function, all the values above 0 are 1, and all the values that are 0 and below 0 are 0 for this type of activation function. So it replicates a little bit about the activation of the cell. There are several activation functions, such as the sigmoids, also the softmax, the hyperbolic tangent, and the ReLU. So we can say that the sigmoid, comprises the values between 0 and 1, while the hyperbolic tangent are between -1 and 1. This can give the networks different kind of approaches. The softmax is the output will be a probabilistic function where will give us where the sum of all the values from a vector will be 1. The ReLU is all the values below 0 will be 0. All the values after 0 will have its own value. Here we have an equation that is logarithmic, and the reason for this is only because to avoid the differential to be infinite or so we can say that artificial neural networks can have several layers, and based exactly on these activation functions. So based on each artificial neuron, so we can have several artificial neurons in several layers. They all communicate with each other, and we can have several layers. And as we increase the number of hidden layers, our networks will become deeper and deeper and deeper. That's why we call deep neural networks. Examples from this, it's the convolutional neural networks. Well, we have the example of the AlexNet that is vastly known in the community of deep learning. That by several convolutions of the image, we can create feature maps only using these weights, and also subsampling. In the end, we will have a vector of features that are learned, where we can attach to a fully connected network or a classifier that will extrapolate those features into an output. Okay, so the idea here is that the network will learn its own weights, and it's going to learn its own filters, so they can have their own. If the humans do not input any information, the network will learn its own features. And as we observe those features closely, we can check that they are pretty close from the ones that the human. Some of them are pretty close as the human created, but others not so much. In the case of recurrent neural networks, we can say that there are several. This is like only a network, a cell with a network cell, but at each state, it's fed with a new value or a new vector. And as the time passes, it will then have new inputs, but it also will have to depend on the state of the previous cell in order for it to learn about the sequence. So if we unravel this kind of sequences, we can have this like in a sheet of paper where everything is like stamped. And when everything is stamped of a sequence, we can say that we have a really, really deep network. And as the sequence gets higher, as this n becomes higher, then we'll have really, really deep networks, and sometimes they are really hard to learn because of the time window that we give into the network. So when using these recurrent neural networks, we really have to make sure that what we put in cannot be that big. So there are two main, I can say that are two main recurrent neural networks that are currently used. So they are widely used. There are others that have been actually gained more relevance, but I will not discuss them. But here we have the example of the long short-term memory, the LSTMs, and the gated recurrent neurons. The LSTM is it's a first version, and gated recurrent units was later made to simplify the equations of the LSTM. What is amazing about it is that before, when we had this kind of networks, the networks would not learn because of something that is called the vanishing gradient that some of you might be acquainted, where the first networks, the first layers would not learn the process. Why was that? Was because there was not an efficient memory, an efficient management of the memory. So the long short-term memory, it has this kind of gates that makes able for the network to forget about certain states or reject the input that is given and only rely on the state that is within. With this, it's possible to learn not only long past things, but also short memory things, right? So in the case of this was widely studied in texts. For example, if we say hello, we need to retain the HELLO. But if we have this on a sentence, hello, I am David Bello, dot, thank you for having me, dot, it will also have to retain the memory not only from the characters before, but also of the dots that happen later on. And this is the long memory. In terms of biosignals, this is also very easily to explain. When we think about the electrocardiogram, we can say that the short memory is like the point right before. And if we say the long, we can say that are the R where the heartbeats happen, for example. The gated recurrent units are just a simplification of the LSTM. Some say that we need less data in order to train them, but it's discussable. So in order for these networks to learn, we have to rely on optimization. So we have loss functions and optimizers. It's very important to say that these loss functions are widely used, the mean squared error and the categorical class entropy. But we as at least me as a time series, I have to be really criterious about these loss functions, because we can not only use these ones, but create our own in order to make sure that the optimizer reacts. So what is exactly an optimizer? Once we have an error function, this is the representation of the two error functions only for visualizing purposes in 3D, where we have several optimizers that descend on the curve to check where the error is minimum. And sometimes this learning process is very important, because sometimes there are some algorithms or even some parameters that we give into the optimizers that will not reach the minimum value of the error function. And I think these two cases depict very well some of the optimizers that can be better for some kind of error functions, others are better for other kind of error functions. Of course, when we are dealing with multiple weights, this is very difficult to visualize, and the only way to see this is the evolution of the error of the loss. Well, I'm going to give you a little bit of the state of the art from 2015 until last year, because last year, in the start of last year, because in the last year and this year as well, there has already been an increase, a lot of increase on the state of the art. So before this, there was only data, human-made feature extraction, and we would only use classification. So most of the papers in deep learning applied to biosignals were published after 2015. There is one remarkable exception, that they used the CNN to extract features and classify from blood volume pressure and electrodermal activity. And it was to classify features into relaxation, anxiety, excitement, fun states. So this was one of the big remarks and this algorithm outperformed human extracted features algorithm. But Martinez et al. Was actually a student of one of the proposals of the CNNs. So, they worked directly with these biosignals. But before, deep learning was using only as a classification later on. So there is two types, where there was data, human-made feature extraction, then we used deep learning for classification. Then I started data, and then there was some kind of data transformation, such as spectral analysis or only watching the ECG peak, sometimes only transforming the signals into images. And then there was some machine-made feature extraction and classification based on images mainly. So usually this was applied to EMGs, EEGs, and ECGs, but also phonocardiogram, where also we had the conversion these signals into images, for example, using spectral analysis, but not always, and uses CNN architectures that were already available, such as AlexNet. So this AlexNet has been used vastly in this field. Examples of applications are the recognition of physical actions through EMG, detection of changes of sleep from EEG, and detection of atrial fibrillation in ECG. So after this, there was a lot of studies where there would only data, some kind of pre-processing, but then we would use deep neural networks to do both the machine-made extraction, feature extraction, and the classification. So this kind of papers now have grown exponentially, and some in the application of deep learning to raw data is more active since 2017. It was actually the year where I started my PhD. And examples using CNN, like in detection of arrhythmias from ECG. And sometimes there are networks that actually are very difficult to replicate, especially in our institutions, since we don't have the machine power to process a 34 layered CNNs. So you can see this was made in the Harvard in partnership with Google, if I'm not mistaken. So there are also examples using RNN, arrhythmia detection with tactile STMs, and the combination of CNN and STM. So I will give you also an overview how this is made. This is actually best results for each application scenario. As I said, synthesis, biometry, and also anomaly detection. And we have some, for example, biosignal synthesis, there are not much things going on. Some of deep learning started using GAN architectures in the last few years. The ECG biometry, now it has been increased a lot, but before, when I did the paper, it wasn't that much used. There is also anomaly detection with, there was no actually found, because the anomaly detection here is not detection of arrhythmias, but detection of things that are not normal. So normally the processing, biosignal processing for deep learning architectures, normally the raw signal, where we have some kind of pre-processing. We have a normalization that is very important, and then we feed onto the deep neural networks algorithms. One part that is very important is the segmentation. So the cutting of the signal into several windows. Sometimes this is very important for the network to learn the process of the learning. So I would say that it's very important to have some kind of pattern of interest, since these networks work better in cyclic signals. But not only, but they work better on cyclic. So it's very important to have a window that at least gets the region of interest. And if we have some kind of overlap, even more, so that the network has a sense that is something that is going on in a continuous way, and learn the different angles of the signal. It's also very important. Unfortunately, this kind of networks, when we don't have that much data, they are very sensitive to noise. So it's very important for us to remove noise. Here we can use only a threshold using means and standard deviations, where we can remove all the signals that are below or higher than that. But we can also use clustering methods that can separate the good signal from the artifacts. Of course, this is important if you don't want to detect, because there are some algorithms where you want to detect artifacts, but even there you have to have some kind of considerations. We also have quantization, and quantization is actually reducing the dimension of the amplitudes. where instead of having not infinite numbers, but like thousands of millions of possible data points, we reduced that to one like 512 or 1000 or 2000 signals, but 2000 steps where the signal different levels of amplitude. This can simplify a lot the life of the networks to learn the different values. So there are also various normalization types that can be used depending on the case. We have the absolute maximum. We can use the amplitude, the standard deviation, the absolute maximum and minimum removal. And we can also adapt this into a window where we can, through the window, a window that crosses the signal, and we do the normalization in the window faction. But this has to be done carefully because it can change too much the morphology of the signal. So I will give you a brief overview to the architectural modules as I conceived. We have the CNN convolutional neural network encoder. Here we have the convolutional neural network decoder. We can have a recurrent neural network encoder and its decoder counterpart. Normally, we have a latent layer after the encoder. What does this mean? The latent layer or latent vector, we can say that is the feature vector, but we do not understand it because of the complexity and the intricacies, because it is not human-made. It was learned by the network. Then we can have a dense layer, feed-forward layer. There are several names for this, but normally it's used for classification. So we can have different combinations of these different modules in order to get a more complex network out of this. I can give you the examples of some architectures. For example, this RNN encoder and decoder, it both encodes and also decodes. And in this configuration, a model may learn to predict the following sample based on the previous ones. This configuration may be used in order to make model learn about the signal's morphologies mechanisms. This configuration only classifies the next sample, as the module does not absorb the complexity of the biosignal with the continuous values. So it's very important to segment in overlapping time windows, but also quantize, because I tried several times not to quantize and it didn't perform at all accurately. The network is fed with time windows and the output will be the same time window dephased by one sample. So if we are trying to predict the next sample, then we give as an input one time window, and then the output will be dephased only one sample, because the first sample will try to predict the second sample, and the second sample will try to predict the third sample, and etc. Here we have a CNN encoder with a dense model. This is widely used, and the convolutional encoder can be used as dense model. So the CNN encoder will encode the features, and the dense module will only give us classification. And then the filters will be learned with this configuration when using one-dimensional convolutional neural network. We can also use autoencoders with dense layers. Now things start to complicate, where we use CNN encoders to learn the features. That is, an autoencoder is the replication of the inputs. So we have one CNN encoder, one CNN decoder. In the middle, we have the latent vector, and the latent vector will be then the features that the network need to have in order to replicate the inputs. Okay, with that feature vector, it can add a dense layer, and then dense module will give us classification. This is especially interesting for transfer learning. Now it can be even more complicated if we add the recurrent neural network to this. So if we have a CNN for different time windows, we encode different features for each time window, and then we add those latent vectors into a sequence. When we have those feature vectors into a sequence, if we feed it to a recurrent neural network, we can have a predictor of the next time window. And with the prediction of the time window, we can also predict what is going to happen next. And sometimes we can even predict if a heart attack will happen, if some kind of arrhythmia will happen in the future before it even happens. There are some other architecture, for example, synthesis using a mixture model where we synthesize a signal based on two inputs, and also generational adversarial networks for domain transfer. So if we have a signal that for example, we only give the information about the R peak of the ECG, it can then make a full ECG signal based on the style. So the application scenario: synthesis, authentication, and detection. So in the biosignal synthesis, we use this encoder-decoder recurrent neural network. And the objective is to teach the network how to understand the mechanics behind the biosignal generation, also replicate it in 3 types of biosignals. So in order to do this, we need to move average and normalize it, have a convolution with Hamming window for it to be smoother, for the signal to be smoother. Then we clip histogram edges and quantization, because the quantization factor is very prone to artifacts. So it's very sensitive to artifacts. So in order to do that, we clip those artifacts in order for the signal to be comprised between 2 values, between 0 and 1. The architecture here is 3 gated recurrent units. After embedded network, we have a regression on softmax nodes, and then we have the output sample. So this is based on a structure that is made in NLP, natural language processing, and we apply to deep neural networks. And here is exactly predicting the next value, and then we predict the next sample. And we did the training with a PhysioNet Fantasia dataset and ergonometer data of someone pedaling a bicycle. We use the optimizer. We also use 30%, 70% training testing with windows of 512, but you can see this later if you want to. And then by choosing the most next value in sequence, starting with a random value, we can predict the next value. Here we have some results, and it's very impressive because we can see that is an ECG, and we can say that here the network had a really, in the second image, we see that the ECG had a really, the model had a really rough time in starting the synthesis, but then after a while, it understood that he needs to regain the synthesis as the real morphology, and he started to replicate the morphology. So we have here the results for the respiratory EMG and DCG, and we can say that the signals are pretty close from the inputs, what is amazing. Sometimes we see a little bit of some glitches, but it's normal to prove that this was made by a machine and not by a human being. And this is the learning stages as the network was learning the signals, and we can see here the process of the learning stages of the ECG. And it's very impressive as it starts as a completely random, and it starts to learn some patterns of the wave. and sometimes it can, and then it start to have some kind of knowledge of the frequency, such as the different patterns and the succession of the patterns, and then it has a notion of frequency between the patterns. From synthesis to authentication. So after developing one, we made an hypothesis. Model A is trained with ECG of person A. If we use this model with person B, then will the prediction error be higher? And then when fed with person A, will the error be lower with the model trained with person A? And after checking this, we have a confusion matrix that is very beautiful. So we started to think about the use of this in ECG biometry. And the idea of ECG biometry is both identification and authentication, where the identification is checking the source of that ECG signature, validation made by accuracy, and the authentication is proof that that ECG is from the claimed owner, right? So validation made by equal error rates. So we can say that all ECGs are different from each individual. It is a signal that all living humans possesses, so it's hard to counterfeit. And using off-person ECG acquisition could be used for high security settings. So we studied two algorithms, the recurrent neural network that we discussed, but we also tried a convolutional neural network with time features. So this is based on dilated convolutions. We feed on two windows. The first one is not fiducial, so we use a time window, but then we check a cycle, and then we give one of the cycles of that window to the other network. After, in the end, we use a fusion layer, where the latent layers are summed, and then we will have the dense layer where we'll classify it. Then the loss is made by categorical cross entropy. So the idea is to have one score in order for us to compare both of them. And we can say that we have scores for the different models and for the different individuals. And we can see that the lower the thresholds of decision, we can see that it classifies better or not. And this is important then to make the ROC curves made for authentication. We tried the Fantasia MIT-BIH and ICBSII. ICBSII is an off-person ECG. And we have here the metrics. So by comparing the error prediction of the ECG, it is possible to have a confusion matrix for identification, and for different thresholds, we can then have a curve. So here we have the accuracy as the batch size. So as the window is higher, the more amount of data that we give to the network, the better results it has. And you can see that here we had really good results. We had really good confusion matrices here for MIT-BIH. And here for ICBSII, it's a very difficult off-person ECG. It's very noisy. It's very hard to give results. So so even though it looks like we didn't achieve that good results, in comparison to the literature, they are very good, especially in terms of authentication. So we can see here that we had state-of-the-art results for these kinds of data sets. And then if we use a deep neural network to learn the default morphology, is it possible to see if it diverges from the occurrence? For example, will it be good to use for noise artifacts and premature contractions? So in this case, we use several scenarios, one with noisy data and the other with arrhythmias. We used an autoencoder to check if it could code the features, and we use those features only made with the clean signals, and we added the signals with noise. And even though it wasn't trained with noise, only the NES network was trained with noise, we could get really good results. Then in terms of arrhythmias, we did a much more complex architecture where we combined convolutional neural networks, but also gated recurrent networks in order to do some kind of classification based to detect the arrhythmias. So the first part, it's also only learns the clean signals, and the idea is to check if the other part of the network could learn the divergence between those features that weren't learned and the output. We got really pretty good results in actually the literature. They had better results, but the literature actually was trained only to detect arrhythmias. And the idea here was to detect not normal anomaly patterns, and then train another network to learn the arrhythmias. So it's a more generalized setting in so if the idea here is if we had more arrhythmia settings, that maybe our network will learn better than the other that was specified only for those kind of arrhythmias. So what was learned? What is the usefulness of these architectures in the future? So in terms of synthesis, we can see that it learns and replicates several signals, and the search of internal neural settlers can be used for morphological aspects of the signal for explainable AI. The authentication showed really good performance margin. There is such a margin for improvements, such as introduction of transfer learning in the training persons and the acquisition of more data per person, especially more angles of the signals. Then we also, for detection, we use the autoencoder for learning normal morphology of a signal, and we can see that it helps in the detection of divergence in the expression of key characteristics of the ECG cycle. So by analyzing and presenting several architectures are providing their usefulness in the research of patterns within the biosignal field, the thesis that I made has shown that deep neural networks has a huge potential in providing new solutions and to the field. And we can see with the exponential growth in the literature in the last 2 years. And some mechanisms of these architectures may be exploited for unveiling how biosignals work and the reason behind the classification of some pathological events. I can envision some future architecture, for example, the fusion between networks using a hierarchical multimodal approach. For example, if we have different networks in the brain, where we had different areas of the brain, and if we do this in hierarchical modes, maybe we would have a better and more informed classification. In terms of explainable artificial intelligence, and I see the visualization of filters through the generation mechanism. So we would, this is already being made. So this vision is already being fulfilled. That is actually synthesizing data with the network that was trained for classification, and then check what happens if we give some kind of input. And therefore it can give us hints of how because normally the network, since we don't understand the latent vectors, they are considered as a black box. I envision the AI in healthcare like a fusion of several networks. For example, if we have a network specialized in the brain, one specialized in the ECG, another one specialized in the X-rays, we can have then some kind of decision support that will have a more informed decision. So I wanted to show the work that I did in FDL, but I guess it's not. I think it will be not possible since we are reaching the end. I wanted to give you some time for questions. So please feel free to ask them. Thank you so much for your time. And thank you for your attention. So if you have any questions, please do so.
Thank you, sir, for your highly informative presentation on the overview of deep learning in the context of biosignals. Thank you for joining us today. As we come to the end of today's keynote session, we would like to extend our sincere thanks to all the speakers of the keynote sessions. We thank the attendees for their valuable time and contribution made throughout the sessions. We hope it was insightful and thought-provoking for all the innovative minds out there. Now we will proceed with the valedictory function. Over to Ms. Supraja.
Hello, sir. Hello, Dr. David. This is Kavita.
Hello. How are you, Dr. Kavita?
I'm good. Thank you. So, students, before you proceed, did you ask for the participants if they have any questions? Dr. David, we were having a discussion today morning, and there was a talk about using LSTM networks for real-time data. So what actually is your opinion about LSTM networks for real-time data? Some people say RNNs are overrated. You don't need LSTM networks, RNN networks. It's enough if you just go in for simple deep learning. So how do you actually distinguish upon what particular network can be used for real-time data, exclusively EEG data? What's your opinion on that?
Okay, so I have actually strong-willed opinions about that. So first of all, I do think that that LSTM can be very useful, not in all kinds of data. So I cannot extrapolate that for all the signals and all the real-time measurements. Currently, in order to make LSTMs a viable solution for using it for real-time inference, only if we have at least some kind of degree of computational power. So if we have a good computer or if we are working in the cloud with a really good computer graphic, I think that maybe the LSTM could provide better results. But nevertheless, I think that other algorithms, such as the ones that you described and other more, can be actually still be used, and I think they are quite viable. I think that in the future, with the exploration, the better exploration, and the creation of new networks, such as currently there are more. There are other networks such as transformers that now are much in the edge. They could be also used on later on real-time inference. So by answering your question, I think it's a yes and no. Depends on the case. But if we are dealing with for example, wireless devices, and we want the inference to do the inference directly. If we don't have some kind of module that supports the calculation, that I will go for the simpler classifiers. That is for sure.
Thank you. So that was very interesting to hear from you. The entire talk was very neat, simple, and of course very informative. And I'm sure the conference. This is the last talk of the conference, and it made a lot of sense because you're ending it on a very high informative note. Thank you so much for joining us, Dr. David. Really appreciate it.
Thank you for inviting me, Dr. Kavita. It's always a pleasure.
Okay, now I think, Supraja, you can go on with your.
Yes, ma'am.
Thank you.
I welcome you all to the validity function of the 8th International Virtual Conference on Biosignals, Images and Instrumentation, ICBSII 2022. I would like to begin the ceremony by introducing our chief guest for the evening, Dr. David Bello. Dr. David Bello is a Portuguese biomedical engineer and a senior scientist focused on machine learning for time series as Fraunhofer, Portugal, and AICOS. David Bello received his PhD in biomedical engineering from the Nova University of Lisbon in 2021 in applying deep learning algorithms for several biosignals paradigms. His research activity was mainly focused on biosignal processing and machine learning to extract meaningful information and make automatic decision support systems. On the industry side, he earned experience in programming mobile applications, reporting services, and administration software. His specialization includes deep neural networks, TensorFlow, Theano, biosignal processing, machine learning, etc. In addition to this, he has participated in the summer of 2019 in the NASA Frontier Development Lab in developing artificial intelligence tools for aiding the astronaut health program. Currently, he is working at Loka Inc. as a machine learning engineer and leader, specializing in the development of machine learning solutions for healthcare. I welcome you, sir. Thank you for making time for us in your busy schedule. I now request Dr. David Bello to address the gathering.
Okay, first of all, thank you so much. Currently, I'm at Fraunhofer, so that part of the biography was outdated. I was at Loka, but now at Fraunhofer as a senior scientist. And I give you a warm, well, here in Portugal is a warm morning, afternoon. I think that this conference, it's very interesting, and I think it should be because people need to be more aware of the things that are being done in the healthcare community. Biosignals, images, and instrumentation are developing real fast. Lots of techniques are being done. And it's a wonderful experience to see how this is growing. And especially on these dire times, as I was mentioning earlier, we can say that healthcare, it's vital. And I can again start by mentioning what I did in the start of my presentation, is that there are several contexts that we cannot anticipate, and it crashes like this COVID has crashes us completely as a society, because we were not prepared to deal in a fast time setting with this kind of impacts. So what I mean is that it's very important for us to increase the quality of our work, do more technical assessment of what is happening, understand biosignals even more, understand imagery on the medical procedures even more, and understand everything related to the healthcare even more, do some real-time solutions, as Dr. Cavity was questioning me. So these kind of approaches are very important for the future, for the present, and also the future. So when something like completely unexpected hit us next, then we will be much more prepared. And so on behalf of the committee of this 8th International Virtual Conference on Biosignals, Images and Instrumentation of 2022. Thank you so much for showing the amazing work that is happening and enlightening us on the state of the art of the things that are happening.
Thank you very much, sir, for reiterating the various approaches towards BME in society as a whole. If we knew what we were doing, it wouldn't be called research, would it? Said
Exactly.
Albert Einstein, and how true the statement is. We hope that this conference was instrumental in pushing you a step ahead with your quest for knowledge for all the participants, supported by the thoughtful questions raised during the sessions. On the same note, we are delighted to release the proceedings of the 8th International Virtual Conference on Biosignals, Images and Instrumentation, ICBSII 2022. Thank you, sir. It is now time to announce the much awaited Best Paper Awards in each of the tracks in the conference. The papers were divided into 9 different tracks, and in each track, the paper was evaluated meticulously by the session chair and session in charges. Kindly give us a few seconds.
Any issues? Maybe you can close the live streaming and then go on with the valedictory, because same thing happened in the inaugural session as well. You hear me?
Yes, ma'am. We thank all the keynote speakers who graced us with their presence for the entire event. And the valuable contribution of all the professors and the student volunteers involved in the event. Now we move on to the winners of the best paper. In track 1, the session chair was Dr. Binoy B. Nair, Associate Professor, and the session in charge was Dr. S. Pravin Kumar. The winning paper ID was number 14, with the name Gender Differences in Postural Stability in a Cohort of Adolescent Age, with the authors S. Varsha, Deepa Advalagan, K. R. Dalarasu, M. Jagannath, and A. Celestine Gerald. Congratulations. Moving on to track 2, the session chair was Dr. C. Vinod Kumar, Associate Professor, and the session in charge was Dr. S. Arun Karthik and Dr. Sachin Gauri Shankar Sarate. The winning paper ID is number 20, with the name Design of an Automated Oxygen Flow Control System for Hyposemic Patients. The authors of the winning paper are Krishna Kumar S., Bethany Jani J., Sindhu Divakaran, Radha K., Monisha S., Lumen Christy V., Cheryl Robinson, and Aishwarya Mohan Kumar. Track 3, which was led by Dr. N. Venkateswaran, Professor, and with the session in charge as Dr. K. Nirmala, had the winning paper ID as number 6, which researched on psoriasis skin disease identification using SVM image classification and determining the growth rate. The authors of the paper are Tamirvani TR, Aparna K. Ajayan, Sandhya V, Hemalata RJ, and Chandrasekharan R. Track 4, which was chaired by Dr. S. Manikandan, Professor and Head, and with the session in charge as Dr. M. Dhanalakshmi, had the winning paper ID as 57, titled Biomechanical Characterization of Human Gait Using EMG Parameters. The authors of the paper are Chitra M. Das, Mr. Nagarajan S., Pungurali, and Mohanavelu. Moving on, track 5, which was chaired by Dr. P.V. Pramila, Associate Professor, and with the session in charge as Dr. V. Mahesh, had seen the winning paper ID as 34, with the title Prosthetic Arm with Functional Fingers and Wireless Recharge on Walk Function. The authors of the paper are Abhijit Bharatan, J. Prem Kumar, T. Sudhakar, Bethany Jani J., and Sindhu Divakaran. Track 6, chaired by Dr. R.S. Milton and session in charge Dr. L. Suganti, had the winning paper ID as paper 48 on detection of anemia using image processing techniques from microscopy and blood smear images, with the authors as Nitya R. and Nirmala K. Track 7, with the chair Dr. Dinesh Bhatia, Associate Professor, and the session in charge as Dr. J. Vijay, had the winning paper ID as paper ID 26, which was researched on computer-aided diagnosis system for breast density classification in mammograms, with the authors as S. Srinidhi, Devi Vijayan, and R. Lavanya. Track 8, which was chaired by Dr. R. Amuda, Professor, with the session in charge as Dr. R. Nitya, had the winning paper ID as number 114, with research on dose response curve using chemically induced PCC assay in peripheral cells irradiated in vitro to 5 MeV alpha particles emitted from radon source, with the authors as C. Meenakshi, S. Chandrasekharan, N.P. Rajesh, and B. Venkatraman. Finally, track 9 was chaired by Dr. Ramya George, Assistant Professor, with the session in charges as Dr. N. Punita and Ms. B. Divya. The winning paper ID of the track was 130, which was a research on low-cost system for recording and monitoring muscle activity through video games applied to children in times of confinement caused by COVID-19. The authors of the paper are Christian Oval, Wilbur Actuazi, Lucas Herrera, Ali Actuazi, Martha Romero, Kajan Rojas, Kitty Urbano, Francisco Hilario, Milner Liendo, Sandra Miza, and Jose Luis Herrera Salazar. A very hearty congratulations to all the authors for winning the Best Paper Awards and much appreciation to all the participants. We would like to open the forum for participant feedback. Interested people may unmute and share their experience with the conference. You may also share your feedback through the Google form link posted in the chat box. Karni ma'am, would you like to share a few words?
Is anyone talking? I'm not hearing.
Yeah, thank you very much.
There's
I
no
would-
feedback. Do we have participants other than SSN? Is there any participants in the meeting here right now? I see one, Ms. Meenakshi, ID 114. Are you available? People are not interested in talking, I guess. That's okay. They, at least we request you to give your feedback in the Google Forms posted in the chat.
Ma'am, Meenakshi is available.
Okay.
Yes, ma'am.
So can we hear from Meenakshi? I mean, this goes like class. We'll have to pick up names from the participants list and then request for talking.
Well, Meenakshi, can unmute yourself and please speak.
I'm just trying to pick up another name.
This is Meenakshi. Am
Hello,
I
can you see
audible?
you?
Ma'am, am I
You are,
audible? Ma'am?
yes, you are. Can we see you?
Yes, ma'am, I could see you, ma'am.
No, we cannot see you.
Ma'am, some network issue, ma'am. Because of that, I have.
No problem. Just go on, please.
Good afternoon to everybody.
Good afternoon.
It was really fantastic international conference, which I have never ever attended. Really, it was very useful, very much informative. Excellent, ma'am. I would like to congratulate all the team members. And this is actually a model for us, like how to conduct this type of conference, that too through virtual mode. Thank you so much, ma'am, for giving me the opportunity.
Thank you, Meenakshi. Where are you from? If you can know, which college are you from?
Ma'am, actually, I'm working in CREST, ma'am, in funded project, IGCA funded project.
That's great. Nice to have you. Thank you.
Thank you so much, ma'am. Thank you, ma'am. Nice meeting all, ma'am.
Yeah, we'll meet in person too. Try visiting SSN when you find time next time.
Yes, ma'am. Sure, ma'am. Sure, ma'am. Thank you, ma'am.
Yeah, thank you. Is there anyone who would like to give your feedback? I have a few other paper IDs reflecting in the participants list. Should I call?
Ma'am, and
Is-
Dr.
is
T.
this,
Sudhakar has raised his hand. So if you'd like to share, he can unmute and
Please, please.
Hello, good afternoon, ma'am.
Hi, good afternoon.
I'm Dr. T. Sudhakar, Head Biomedical Engineering, Satyabama Institute of Science and Technology.
Great, sir.
It was a wonderful organization. Really, I must admit, amazed the way you people have started and completed the conference. Wonderful, ma'am. No words to praise. Really, I can take it as the next time you're going to organize a conference. I can take this as an example. It's excellent. It was excellent, and the way the keynote speakers and everything excellent. And we are very happy that we have banked 2 prizes from Satyabama. That also very happy. Really, it was wonderful organization. Really, and the journey people like Scopus also. Thank you. Thanks a lot.
Thank you. Thank you so much, sir. There's one Ms. Anjana Dwivedi raising her hand. Can you just help her out?
Yes, good afternoon, everyone.
Hi, good afternoon, Anjana.
Yes, ma'am. We are really thankful for giving us this opportunity to be a part of this conference. And I really felt that this conference was very informative and also like it was very well organized. And without wasting a single minute, we have utilized every minute of the conference and we have learned a lot from this. So thank you. And I congratulate every team member for organizing such a wonderful conference. Thank you.
Thank you so much.
thank you very much, ma'am. I would now like to invite Dr. Geetanjali, Associate Professor in the Department of Biomedical Engineering, for delivering the vote of thanks.
Ma'am, you are in mute, ma'am.
Can you able to hear me?
Yes, ma'am.
Okay. Sorry about that. It has been a great pleasure for us, the organizing team of ICBSII 2022 Department of Biomedical Engineering, to host all the participants of 8th International Virtual Conference of Biosignal Images and Instrumentation. On behalf of the entire organizing team of ICBSII 2022 and the Department of BME, SSNCE, I extend my sincere vote of thanks to all the speakers and participants who made this conference a grand success. We are very grateful to the management of SSNCE, President of SSN Institution, Dr. Kala Vijayakumar, and the Principal of SSN College of Engineering, Dr. Annamalai sir, for granting us this opportunity. We thank Dr. Shashikant Albal, Director of SSN School of Advanced Software Engineering and Career Education, for his timely support for conducting the online conference. We are very thankful to the head of our department and organizing chair of ICBSII 2022, Dr. A. Kavita, for being the pillar of support and for all her advice, which greatly helped towards successful organizing this conference. I sincerely thank both the international and national members of the advisory committee for their valuable suggestions in organizing this conference. We have been fortunate to have many eminent speakers across the globe from academia, research and development, and industry working in the field of BME. We sincerely thank our chief guest, Professor Ajit Yoganathan and Professor Joshua Evan, and our chief guest of honor, Dr. Srinivasan Ramamurthy and Dr. Mauganavelu, for kindly accepting our invitation and gracing the inaugural ceremony of ICBSII 2022 with your virtual presence, pearls of wisdom and knowledge. We are truly humbled by your presence and thank you once again. We also sincerely thank Dr. David Bello for accepting our invitation to be the chief guest of our valedictory function. We extend our heartfelt gratitude to all the international and national keynote speakers who joined us from the USA, the UK, Portugal, Kuwait, Malaysia, Singapore, and from various parts of India, who took time out of their busy schedule and motivated the participants and audience with their rich insights on the multidisciplinary fields under BME. We would like to thank all our technical committee members of ICBSII for their continued support, valuable comments and suggestions they provided in reviewing and further enhancing the quality of the papers submitted. We thank all the technical chairs, publicity chairs, finance chairs, conference registration chairs, and publication chairs of the Department of BME. We extend our special thanks to Dr. Sachin of BME Department for helping us stream the conference live on YouTube. Furthermore, we would like to acknowledge and thank the audience who remained patient throughout the event. We have also made sincere efforts to enhance the quality of the received paper on our part. This year, we received 139 papers totally, among which only 56 papers were accepted after carefully double-blind peer-reviewed of anonymized papers. Only those peer-reviewed accepted papers with similarity index less than 12% adhering to the strict guidelines of the Scopus Index Journal of Physics Conference Series will be forwarded for the possible inclusion in the Journal of Physics Conference Series IOP publisher. The end of a story has always led to new beginnings for many of us. Although ICBSI 2022 ends here, we are sure that the lesson learned from the renowned speakers from day one of our inauguration till the last session will certainly help the academician, research scholars, and the students to take a great leap forward in accomplishing remarkable and innovative research findings. We, the Department of BME, Assistant CEO of Engineering, will continue to conduct ICBSII with the support of enthusiastic participants like you all in the forthcoming years. On this note, we conclude this memorable event, the ICBSI 2022, by thanking everyone whose contribution have made this conference successful. Have a wonderful new beginning of researching. Thank you.
Thank you very much, ma'am. Let us use this gathering to take a moment to remember Dr. Bhagiraj. Over to Padmavati to speak a few words in his loving memory. Slide மாட்டீங்களா? மாத்து. Dr. Bhagiraj sir took 2 subjects for our batch, electronic devices and circuits in the first year and diagnostic and therapeutic equipments in the third year. He was not only a knowledgeable person, but also a hardworking individual. I had the opportunity to work under him for our mini project on stress detection using Arduino with 2 physiological parameters. He was always so approachable and down to earth and even suggested to use other parameters. I still remember his words to me. It was, no issues. When you all come to college in April, we will do the GSR part in the DTE lab. He was known for his humility and punctuality, and I have never seen him come late to the class. His last words to us was, "I'll see you guys in the next class." But sadly, we could never see him again. I would like to conclude by saying that words can't fathom his work and his contributions. The last but not the least, we all miss him so much and will hold on to the memories and lessons we learned from him.
Okay, that happy note suddenly moved all of us into immediate tears, and it always happens every time we think about Dr. Bhagiraj. And like you students, we all miss him very badly, and every moment, keeps reminding us. Even afternoon when we were taking a photograph, Dr. Mahesh was mentioning, like, I'm not interested in a selfie anymore, because the last time when we had a selfie, we had him. So he's still with us, and he'll always be with us. And yes, I mean, probably you can take a lot of morals and values that he's been teaching to us, and he'll be happy to see us, develop into very individuals with a very strong discipline and moral values, like how he is. He's been an excellent human being, other than the fact that he was a very organized and disciplined teacher. As a friend, we all miss him, and we will continue to miss him, because he's someone who can never be, forgotten. And I'm happy that you guys remember him, and you actually, made this as a tribute. It's a real tribute, and every event that we will be conducting, especially ICBSII, because when it happens in person, he was the one who roams around every here and there, bringing in people, organizing stuff, staying late nights with all the chief guests and other guests who come over here. So it's very obvious that we'll be missing him all throughout our life. And to Dr. David, who is here, he might not know Professor Bhagiraj. He was a colleague and a very dear friend of ours. We lost him in COVID last year. So it still is a pain to all of us, and that is what our students had. They were kind to pay a tribute to him today. So that is what it's about. And yes, moving on. On a personal note, I would like to take this opportunity to really congratulate the entire organizing team. The organizing chairs, Dr. Geetanjali, Dr. Subhashini, Dr. Saranyan, Dr. Paulan, who were very instrumental in keeping up the activities intact, and managing such a big crowd of participants, and events, and speakers, and everything was organized so well. And student volunteers were amazing. Everything was going on so smoothly, and the continuous expert talks were the highlight of this particular conference. We never had any break. We had amazing expert talks till the last one that we had from Dr. David. So that was a very nicely organized, as the participants mentioned. It was a very, very wonderfully organized and very resourceful conference. As always, ICBSII has been a very big success, all the 7 episodes previously, and this 8th one is no less than anything. So we're so happy that it has come to an end note with a lot of meaning and a lot of success stories to share on. Next year, hopefully, we will have it in person. And Dr. David, we will pull you down to India next time. So please reserve a slot so that you can visit us in person. And my hearty congratulations to each and every one of you, all the students. Suprata, you were amazing. You will be our host hereafter, I guess, for most of the events. So that was very nice. I really wanted to congratulate Tuhina. I wanted to congratulate Yamini, Anjana, and Rebecca, Chandramouli, everyone who were actually the voice behind the conference. It was very good, and you all did a very good job. Thanks for being so interested and supportive in this entire event. Thanks a lot from my side, and on behalf of the management and the BME faculty, I would like to thank each and every one of you who made this event a very, very beautiful, memorable, and successful one. Thank you.
Thank you, Mom. Thank you. I once again thank everybody who is for taking part in the 8th International Virtual Conference on Biosignals, Images and Instrumentation, ICBSII 2022. And we hope to see you in the next edition, 2023. Thank you.
So do we have our national anthem?
I would
Yes,
like to
ma'am, we are.
request the audience to rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you everyone for joining. God willing, we will all meet again for ICBSII 2023. Take care. See you.
Thanks, Colin. Thanks a lot.
Thank you, ma'am.
Thanks, Dr. David. See you soon. Bye-bye. Have a good day.
Thank you, Dr. David Bello. Bye-bye.